very nice introduction. So, biointerface science is what I'll talk about today. My research has two areas, one where we use molecular biology to make materials which are amino acid-based polymers, unstructured polypeptides. We do a lot of different things in biotech and in drug delivery. I will not talk about that today because it's not as relevant. The other area of my research is biointerface science, which is nothing but how to play with biomolecules at some interface, whether the interface is a cell membrane or in this case, a glass slide. And I'll show you what happens when you start playing games with proteins at surfaces. You accidentally end up in global health. So everything I'll talk about today is an accidental journey. Most of the most interesting things in my life have been accidents, and I plan to do things, the outcome has been relatively trivial and boring. So for many years I was interested in this question. I wasn't the only one. Why do proteins adsorb to a surface? Why do cells adhere to a surface? I mean, some of the physical forces are known, but really the flip side, the corollary was, how can we get rid of it? And this is actually a non-trivial question. Getting rid of protein adsorption or cell adhesion to surface is almost impossible, but I'll show you some technological solutions. A solution we came up with that launched us on this journey towards first just diagnostics, then point-of-care diagnostics, which then led us into this area of global health. In general, the answering this question of how to get rid of protein adsorption is important for biomaterials. I also did my PhD in the biomaterials field, and people were trying to figure out the answer to this question. Because if you can get rid of protein adsorption at an implant that's in the body, you can sort of prevent many of the unfortunate events that happen to implants in the body. And once you get rid of all protein adsorption, you can then re-engineer in specific receptor-ligand interactions, and then maybe try and promote healing rather than implantation or corrosion or whatever else is going to happen. It's also equally important actually in the area of sensors and clinical diagnostics, but has largely been ignored because, as I'll show you in diagnostics, which is often done on a surface, what you're looking for is high signal to noise. And if proteins stick to a surface, that's where you get most of the noise from. So the entire area of diagnostics is largely focused on saying, I want high signal to noise at low concentration. So my signal is going to go down, let me amplify it. But when you amplify a signal, you also amplify noise typically. And it's sort of, there's a threshold, there's an asymptote of the kind of sensitivity, low limits of detection you can get by amplification methods, especially when you're doing it on a surface. And what I'll show you is the reverse. We focus on noise because the idea was, if I'm looking for high signal to noise, and I know my signal is going to drop as concentration of the analyte drops. It's a given. If noise drops to zero, I will have infinite signal to noise. Well, you can never get zero noise, but I'll show you a system that has very close to zero noise background adsorption, which is the largest source of noise in clinical diagnostics, and what happens when you start getting rid of noise from a surface. I know this all sounds maybe a little too abstract, but you'll see it over and over again. I'm just sort of foreshadowing it. There were a few solutions. I won't go through them because you have to be interested in biointerface science or surface chemistry and so on. There were a few solutions when I started, but they were extremely limited. One of these was something called cytosol monomers in gold. They only worked on gold and other noble metals. Gold is not that interesting as a biosensor surface unless you're doing a very specific class of sensing using surface plasmon resonance or particle crystal microbalance. What you want is a technology, a solution to get rid of protein adsorption from any given surface, where glass would be really important. A lot of things are made on glass, and plastics. That's the two kinds of surfaces, the two kinds of materials you'll be using to build chips for diagnostics. So when I got into this field, it was well known that if you take a synthetic polymer called polyethylene glycol, most of you know PEG, it's actually everywhere. It's in shampoos, conditioners, it's even in mayonnaise, I believe. PEG is an inert polymer, so the body is supposed to tolerate it well. It turns out if you take PEG and you put it on a drug, this is where most people know it. A lot of biologic drugs are PEGylated. You put polyethylene glycol on a protein and you suddenly extend its half-life. It stays in the body much longer and the drug is more active. A lot of biologic drugs are PEGylated. The same thing happens on a surface. When you put PEG on a surface, what you're really doing, which is the same thing that happens in the body, you're preventing proteins from binding to whatever PEG is attached to, whether it's a protein or a surface. So if I attach PEG to a surface, I can reduce protein adsorption. This was known from the mid-1980s, and people then spent 20 years trying to figure out how to really make it work well, because when you attach polyethylene glycol, which is a linear polymer, to a surface, you will reduce protein adsorption, but you can never get rid of it because you have to, the way people were doing it, they were synthesizing PEG in a test tube, in a beaker, and then they were attaching it to a surface. What they really needed and wanted was a forest of PEG on the surface, but the analogy I use is It's like trying to plant a forest by growing up saplings in a nursery, uprooting them, putting them on a helicopter and throwing them at the surface. What you get are clumps. You never get a complete forest. If you want a forest of PEG, because once you have a forest of PEG, proteins will approach the surface, they'll see the PEG, they have no energetic reason to bind. So thermodynamics are not favorable, and they will simply sort of stick and then leave, get desordered. If you want a forest, do what nature does, plant seeds and grow trees. And if you're going to try and grow a forest, what you want is not conifers, you want deciduous trees with large branches and a big footprint. So PEG is like a conifer, like a pine tree, it's a linear polymer. It doesn't have much of a footprint, so you have to pack linear PEG at a very high density, which is kind of difficult. If you want to plant seeds and grow trees, it's better to have the small seed and then grow a tree with a bigger footprint, so I don't have to plant as many seeds and the process becomes easier, and that's exactly what we did. So we took this technology which had already been invented, which is called surface-initiated polymerization, where you attach on a surface a seed, a chemical group which can interact with a monomer, a building block of a polymer, and will grow a polymer only at the surface. So, and you want to do this in a controlled fashion, so there's a technology called atom transfer radical polymerization, which allows you to sort of attach monomers step by step very carefully and have very well-defined trees, which are all approximately the same height. And if you select the right monomer, the right building block, you will end up with this sort of tree-like architecture, where the backbone is not PEG. The backbone is actually alkane, it's greasy, but the branches are short polyethylene glycol. So think of them as branches where the leaves are short PEG chains. This is a different architecture from linear PEG because the PEG is not distributed as short branches. And the question was, A, could we do this with a monomer, with a building block that contains PEG? So we simply bought this monomer, oligoethylene glycol methacrylate, and we grew this polymer, oligoethylene glycol methacrylate. To plant a seed on the surface, we first started on gold, where we took a molecule where one end, this thiol group reacts with gold, and this other end is the seed, this bromoisobutyrate group. This is the seed that, when the monomer interacts with it, will start polymerizing and growing the tree. We also did it on glass. That's the second experiment we did, where the anchor now is a silane group, and it's the same molecule. So all you need to do is find a way to attach this molecule to a surface. On gold and glass, you can do chemistry. On plastics, we have many other tricks, and we've done it now with many, many different plastics. We've done it on polystyrene, polyethylene terephthalate, which is what your bottles are made of, polyethylene, and so on. This method is very simple. All you have to do is attach the seed, throw in a bunch of chemicals and your monomer, and it'll happen at room temperature. And the growth of the polymer is linear with time. So you can just pull it out, rinse it, and you stop polymerization. And we've done this now in many, many different polymers. You can do this in alcohol-water mixtures, so it's fairly environmentally benign. You could use, you could actually grow it in scotch if you wanted. You could grow it in whiskey, it can grow. It works as a solvent. And it gives you a very, very thin, what's called a polymer brush, a very thin coating. Our thickest coating is about 100 nanometers. That you can't see 100 nanometer coating, right? The human head is about 100 microns, so extremely thin. It doesn't alter any of the optical properties of the surface you're working with, which becomes important when you're trying to do detection, right? So if you want to preserve the optical transparency of glass, it will be completely preserved because 100 nanometers is less than the wavelength of light. So how does this work? It works shockingly well. So many times I had this idea that this should sort of work, but I didn't know how well it would work. And this is really very old data, now 15-year-old data, which was published in Advanced Materials in 2004. But the student came back and showed me some data where he made a series of coatings. Zero means no coating. It simply means you have gold and you've attached an initiator, but didn't do the polymerization. That surface is pretty sticky, and what you're looking at is something called surface plasmon resonance spectroscopy, where you shine a beam of light and you look at the change in change in refractive index at the surface. But the physics behind it is not important. What matters is that this signal on the y-axis is linearly proportional to amount of protein bound at the surface. And it's not the entire signal, it's this steady state signal here. So this is baseline, you inject protein, you see a large change in signal. That change in signal comes from both protein binding to the surface, but also because you've gone from buffer to protein. Protein has a different refractive index, so you get two contributions. To figure out what's bound at the surface, you then wash it with buffer again, and you come back to the same optical density of the solution, and this different signal. This signal is proportional to the amount of protein bound. So you can see lots and lots of protein bound. When I took gold, attached the seed, didn't grow a polymer. And then I put seeds at different densities on the surface and asked the question, at what density would I start to see a big effect? It turned out at even 5% density of the surface, I reduced of the seed. I reduced protein adsorption a lot. By the time we had 10% density, and beyond it didn't matter, because at 10% we had seeds close enough that the branches were starting to overlap with each other, and when proteins came down, they could simply not find any holes in the sort of canopy in this tree and penetrate to the surface. So this was exciting because, gold is easy to work with actually. Gold is one of the best materials to do chemistry with. It sort of sounds odd if you don't do chemistry. But we knew that glass was going to be tougher. It's tougher to functionalize glass, and plastics would be even more difficult. So it was important to find, this result was important because it told us I didn't have to do a very good job of planting seeds on a surface. They could be fairly sparse on a surface and still get a protein resistant, also what we call a non-fouling surface. We then next did it on glass. On glass, we had a surface where we attached the bromo group, the ATRP initiator, and you can see lots and lots of protein sticks. This is lysozyme, fibrinogen, BSA, and this is undiluted fetal bovine serum. So concentration, this is hundreds, if not thousands of proteins from serum at 60 milligrams per mil. So it's a soup of proteins. In any case, all of these are absorbed, but the moment I made a brush that was even more than, that was about 10 nanometers thick, I saw no protein absorption. So if the brush is 10 nanometers thick, no protein absorption. At 100 nanometers, the same thing. In fact, we found over and over again that if you want to make a good brush, all we have to do is polymerize it, measure the dry thickness. If the dry thickness is 10 nanometers or greater, it's going to work, which is kind of a nice engineering rule. Shoot for 10 nanometers or greater, and it will be fine. And also you can see an uncoated surface, lots and lots of cells sticking, coated surface, no cells stick. So now we have a surface which is protein resistant and cell resistant. The question is what to do with it. And we could have gone down two paths. One is the path, so putting it on the path of putting it on implants, putting it on the body, and I decided not to do that because I did my PhD in a biomaterials group, where basically everything failed. Right? So people got PhDs for things that didn't work, but published some papers. It was sort of somewhat depressing. The body is sort of the biggest enemy to technology, right? The body is evolved to fight against things you put in. So I decided I'd take a simpler path and work in an area I knew nothing about. This is sort of how my whole career has gone. I learn as I go along. It's my ignorance that sort of drives me to learn, and my students teach me. So it's a great, people think professors teach students. I have the reverse. I have a great job because I have a job and well paid, but my graduate students actually teach me as they go along. And at the end of it, I pretend to be an expert, but I'm really not. This is the truth of academia. One truth. There are many truths. So I decided to go into an in vitro field of in vitro diagnostics because I thought, it's in vitro, it should be easier than trying to put this surface back into the body. And so, the question is, there are lots and lots of work done in clinical diagnostics and lots of papers, even in Science and Nature, people stop at working in buffer, which is completely useless because, getting something to work really well in buffer doesn't tell you anything, whether it'll work in mucus or blood, the really challenging environments. And we thought we wanted to work in blood because we thought blood is the most difficult environment. Mucus is also difficult for different reasons. So we wanted to assay directly from blood. We knew that we didn't want to do anything where we couldn't work in blood, and we didn't want to sacrifice sensitivity. So when you work in buffer, you get great limits of detection, very low, and then you go to blood, and everything goes to hell, and things, 100,000 times worse. Or they don't work at all because everything clots and clogs up your device. We decided to go into diagnostics because we thought the fact that we can get rid of protein adsorption and cell adhesion meant that we would have a hope of working in blood because we have a very clean surface. Even if cells sort of settle on the surface, we should be able to gently displace them. And then, of course, if you want to build good biosensors, you need good markers and you need good detection reagents. We did not want to reinvent clinical diagnostics. So, one of the most powerful technologies that was invented in the 1970s is a sandwich immunoassay where you put an antibody down, it captures an analyte, and then if you are smart enough or lucky enough, you have a second antibody that binds at a different epitope, different region, which then binds to half of the sandwich and brings in some signal, an optical signal like a fluorophore or an enzyme that can be converted to produce lots and lots of color. Okay, so everything I'm going to talk about today is a sandwich immunoassay, simply sort of enhanced, redefined, reimagined, if you will, in the rest of this talk. I won't talk about these things. I want to presume that we have good markers. There's a whole world of biomarker generation, people who just find biomarkers doing proteomics, genomics, and so on, and the people who design antibodies, and we're going to rely upon that. So the first thing I said was, let's use this to make a sandwich immunoassay. I had a student who developed this technology, and if you ever go to his PhD, his last chapter is actually, he tried to use it for a microarray. What he did was, he took this surface has this brush has hydroxyl groups at the end. So he activated the hydroxyl groups to make them very reactive by doing some chemistry, and then he inkjet printed the capture antibody to try and covalently capture it, because that's the obvious thing to do. If I want to build a microarray, if I want an antibody to be able to surface, and my surface is protein resistant, it's probably a good idea to chemically couple it to the surface so it doesn't float away. When he did that, everything went to hell. We got horrible detection limits, and he wanted to publish a paper, and I simply refused. I said, no, I think this technology is worth more than this paper that looks as bad or worse than anybody else's technology. So I killed that last chapter as a paper. It never got published. I had a new student who came in, he's one of the most intuitively inventive people I know, Angus Hucknall. He sort of messed around for a year and a half, and he came back with a solution that got rid of this sort of poor performance of a first generation microarray. Instead of taking the surface, activating all the hydroxyl groups, then inkjet printing a spot of antibody to couple it, what happens is you then have to deactivate the background, right? Because it has all these reactive groups which will attract proteins from blood, which is why we had poor performance. The problem is that the deactivation step never goes to 100%. So even after you deactivate the surface, there's still some residual reactive groups, and then your antibodies come down, reagents come down, and they stick everywhere, and you get horrible signal to noise. He figured out that you could take this polymer brush, you could dry it down macroscopically, put in a desiccator, and then he would inkjet print using a non-contact printer, just piezoelectric inkjet printing. After printing the spots of the captured antibody directly on the brush, no chemistry, he would put it back in the desiccator. These are all like really sort of counterintuitive things to do. When we tried to publish this paper, we were rejected from every major journal. They said, you're desiccating your antibody after you print it. Well, it should be dead. We think this is all an artifact. The results are completely wrong. They're an artifact. How could it work? You're inkjet printing onto a non-fouling surface, and protein-resistant surface. Why do you still have spots when you do the assay? The moment blood hits, the antibody should dissolve. I won't go into the details. It works, and we sort of understand why. But very briefly, when you inkjet print on a dry brush, that dry brush has a lot of PEG. It loves water. It actually sucks the antibody in. And you still have a little bit of liquid left. And then when we put it in the desiccator and evaporate off the water, the question would be, why is the antibody not denaturing? So the antibody is physically getting entrapped, which is why you will see spots when you do an assay. It doesn't bleed. It doesn't get all streaky. And you get spots, and you get a dose response curve, meaning this works. Otherwise, you wouldn't get a dose response curve, because even though I print on a dry brush and then I dry it again, you can never get rid of the interfacial water from a polyethylene glycol brush. Just like with the protein, PEG binds water with very high affinity. When you put it in a mild vacuum, you just pull off the bulk water. You still have a lot of interfacial water, and proteins only need interfacial water to survive. This is sort of well known. Okay. So just to show you how this works, we printed captured antibody spots. Okay. These are all the same, and then we would incubate it with the analyte. Then we'd wash it off. Then we would add the detection antibody with the fluorophore. Then we'd incubate, wash it off. So that's typical sandwich immunoassay, but just being done in these microarray spots. And what you get is a dose response curve in blood that in the best case is in the femtomolar regime, which is pretty high, right? Sub-picomolar. We're talking things like less than picograms per mil for those people who work in grams per mil units. So this is a very simple technology, because the fabrication of this microarray is just basically make the brush, which takes 5 or 10 minutes, rinse it in ethanol, dry it down, go to your inkjet printer and print. Now you could use a normal inkjet printer. We happen to use an expensive inkjet printer optimized for proteins, but you can actually rewire your favorite inkjet printer for $200 that you buy and change the cartridge and put your proteins in, and you can inkjet print. It actually works. So this is a very simple way to make an assay. There's no covalent, so there's no chemistry, it's just printing. And as I'll show you, that once you print the antibodies, they'll live and die there, they'll live there for years, because that PEG preserves them. Because remember, how do we store proteins in biochemistry labs? In ethylene glycol, which is antifreeze. This is polymeric antifreeze. So proteins love being in this polymeric antifreeze environment. And you can see how clean the data is. This is raw data, so you can see actually spots even going down to 0.1 picograms per mil here. You can barely see it, and at 1 picogram per mil. The background is completely black. These are not massaged. There's no data processing here. This is what the data looks like. So this is just fluorescent micrograph. You take a fluorescent picture, and the intensity is proportional to concentration, and you can actually build a dose response curve. So at this point, we sort of realized we had accidentally stumbled upon two things. One is that we were working in blood, which is that's when the bulb went off. He's like, maybe we can do point of care because we're working in blood and we're working with finger stick volumes, which we weren't doing, but we were working with microliters of blood because these spots are only 150 microns in size. So it doesn't take a very big droplet of blood to cover the entire microarray. And we should be able to also do multiplexed arrays because I can print different capture antibodies in each column. And I could do multiplex point of care assays, which is sort of a bit of a holy grail. There are not that many good multiplex point of care tests out there. So we went back and decided to I made up my own list of questions. What attributes should a point of care assay have? This was sort of my naive list. I said it should be easy to manufacture because I'd like it to be manufactured not just in the US, but across the world. And if it's easy to manufacture, it is more likely to be cheap, and cheap is really, really critical. A chip that costs $1 is too expensive for many parts of the world. It should be, manufacturing cost should be cents, less than 10 cents, or a cent or two. In our case, the largest cost of manufacturing is the cost of the antibodies because we buy them in very small volumes from suppliers. Once you start making your antibodies, the cost drops to nothing because you're inkjet printing picoliter volumes. So even when I buy an antibody of hundreds of dollars and I get like 100 microliters or 10 microliters, I can make many chips. And I'm paying enormously exorbitant prices for it from R&D systems and Santa Cruz and so on. If you make it, you can make 5 grams per liter of antibodies these days. I mean, the technology is there because antibodies are drugs. The drug industry's figured it out. And for it to be a point of care device test, all the reagents by definition have to be in the device. So if you look at sort of the point of care tests that have been approved, like the iStat for troponin and so on, and you open it up, it looks like a baseball bat, you open it up, it's essentially a large clinical analyzer squeezed down into a small device. It has all the same kinds of parts. It has pumps, valves, tubings, because the whole point of a point of care assay is that you have to keep reagents separated in space and time and bring them together at the right time, if you think about what a point of care test is. So the conventional way would be to say, okay, I'll have I'll have a chip, I'll have the capture antibody here, my detection antibody will be in this pouch or here, then I'll have a pump and valves and tubing, and I'll just do all the pumping and washing steps in my device. That's one way to do it. Uses classical engineering. We don't want to do it. It's been done, and we're not very good at that kind of stuff. Industry is better at building these kinds of devices. So this was a challenge we had to solve. We had to be able to put all the reagents on the chip, but we wanted a simple solution. And this technology should be robust. Once you sort of have your reagents or your chips for the assay, you'd like them to be shipped and you'd like them to have a long shelf life because you don't want the reagents to be replaced constantly. In many parts of the world, that's not feasible. You need fast readout. Our limit is 5. We can do some proteins in 5 minutes, but he said 1 hour is probably okay. It's not great, but if you really want if you want to go down to very low detection limits, you have to go to longer times. There's no getting around that. And multiple analytes, which we already knew we could do. We want to work in complex mixtures, which we could do already, and we could already work with small volumes. And the final part was how you're going to read it out. If it's point of care, well, the best detector, the most ubiquitous detector is the human eye, which is what you use for the most popular, probably the most elegant point of care ever designed, which is the lateral flow assay. Also, in America, people know it as a pregnancy test or the blue white test. It is, I think, a brilliant design. It's simple, but it's fantastic. And the readout is with the eye. That's one of its most powerful attributes. But that makes it qualitative or semi-quantitative because the human eye is not that great at interpolating beyond a certain level of sensitivity. So we said, if you want to, since our assay is so sensitive, there's no way you could use the human eye for it. So we said, well, the next best detector that is getting to be ubiquitous is a smartphone. And you might argue that it's not really true. Smartphones are still expensive, but the price is dropping. And it will keep dropping, because this is electronics and everything keeps dropping. So, and it's not as if every patient is going to have their own smartphone or smartphone-based detector in Africa or India, so the doctor will have it or the clinic will have it. If my cost is on the order of few $100, it's possible to have tertiary care clinics and sort of those roadside clinics, to own one of those devices. In fact, you could probably give it away. So here's sort of our solution. It was work from finger stick blood, from a finger, which is like 30 to 50 microliters. We already had that figured out. We already had figured out this whole problem working directly in complex mixtures. So I did not want to do any pre-processing. In many devices, you take blood and you filter through to get serum or plasma because the blood is gunky and so on. We didn't want to do that. We wanted to keep the test simple. We know we can already work in blood because what we can do is we can print some heparin on the chip separately. And then the blood dissolves the heparin and it doesn't coagulate. The challenge to solve was how do we solve this problem of having all reagents in this device? So Angus is really smart. He kind of was inspired with the LFA and put all the reagents on the chip itself. The LFA works by the principle of capillary flow, but we don't use that here. Instead, we bring everything together in time and space at the right time with diffusion along 2 dimensions, along the polymer brush. And I'll show you how that works. It will be reminiscent of the LFA. And so, how does this work? So here's the glass slide. We grow the polymer brush, then we're ready. Then we print the captured antibodies directly into the dry brush. This takes seconds. You print spots. These are not labeled with the fluorophore, so you actually can, in the central region, you would not be able to see anything. You can actually barely see the scattering from the salts in the buffer when you print. If you want to build a single analyte, you just print 1 column or 1 row. If you want to multi-analyte, you can print different rows. Each row would correspond to a captured antibody for a different analyte. Okay. And I'll show you we've done up to 6 analytes now, which is quite a lot. Then I need to have a detection antibody on the same device, meaning in this case on the same chip. And when blood hits, I need to liberate it so that I can complete the sandwich, right? So the way he did it is he took the detection antibody and mixed it with a large amount of excipient, something that would dissolve with blood, so that when blood hit the detection antibody spots, the whole thing would dissolve. So you print captured antibody from buffer, but you print the detection antibody by mixing in 10, 20, 100 molar excess of your favorite excipient, which could be PEG, which could be trehalose, which could be any excipient. And the idea is that when the assay is completed, these should dissolve, the intensity of detection antibody should go down, and then you should see bright spots emerge in the central region if you completed the assay. That was a first generation assay where we actually mixed the excipient with the detection antibody and printed these spots. We now do it slightly differently, where we first go the polymer brush. You're looking at it in cross section. This would be a spot of captured antibody. Then we do a line scan of trehalose at high concentration to make a pad. We print a pad of trehalose, and then we go back and print the detection antibodies on the trehalose pad. We do it all with the inkjet printer. So we call it the D4 assay because we dispense a drop of blood. The blood dissolves the trehalose, which then liberates the detection antibody. Both the detection antibody and the analyte diffuse and complete the sandwich, and you will see spots appear. This is the kind of data you can get. This is our sort of best array right now, our most advanced array. This is 6 different antibodies. This is kind of not a fabricated sample, but this is a sample where we had captured antibodies against 1 analyte, 2, 3, 4, 5, 6. And we took all 6 analytes at high concentration, mixed them up in buffer, and then exposed the surface. And you can see you get this really high signal to noise because you have high signal, very, very low noise. So let me just show you sort of our best assay we ever built, which is for IL-6, because you can get the best antibodies against IL-6. And what you see here is in serum, a dose response curve, and in blood, they're actually remarkably similar. So, and I don't even show you buffer. So you go from buffer to serum, you lose sensitivity of a factor of 5 or so, but from serum to blood, there's not much of a loss, and we get a limited detection of 100 femtomolar. This is the best array we've ever built, best point of care assay, because these are the best antibodies you can get. IL-6 happens to be the best pair around that you get from R&D systems. And here's what the raw data looks like. So here's this one chip. This will correspond to 1 data point on this dose response curve. So you can see this is a control spot where we just print PBS, and here we printed the capture antibodies against IL-6. When you complete the assay, you can see a bunch of the detection antibodies dissolved, and you get these bright spots. So you take each chip, expose each chip to a different concentration, quantify it, and you get this entire, that gives you the entire dose response curve. This just shows you a 3D reconstruction of actually a single spot to give you a sense of the signal to noise. Here's when you expose to blood on the brush. You can just see little noisy spikes. This is just electronic noise. At 375 femtomolar, you can start to see the signal emerge. At 12 picomolar, 190 picomolar, you actually saturated the assay in the picomolar regime. This is that sensitive. This assay happens to be, again, very serendipitously, extremely robust. It doesn't need a cold chain at all. Once you print the capture and detection antibodies into the brush, you can leave them for 3 months and come back and look at another chip. So we did one assay on day 1, on the day we printed. We left it at sort of Duke laboratory conditions for 3 months. Absolutely the same performance. And somebody said, well, you know, Africa is not like Duke at 20 degrees or whatever, you know, I'm always freezing at Duke. It's going to be 45. What about 45? What if even in the US your FedEx truck broke down? Or what if in Africa, something happened in Africa? So okay, I said fine. 45 degrees Celsius versus ambient at 24-hour storage, 45 degrees Celsius, 120 hours. You fry, pretty much frying at 45 degrees Celsius. So this is baking in some truck, you get the same performance. So we get, if things break down and stuff is exposed at 45, which is not desirable, it still lasts for 5 days. You probably don't want to store it at 45, so some minimal refrigeration or store it in the dark is probably desirable. But you don't need it at 4 degrees Celsius, and you don't need it to be hydrated. It can be stored dry. In fact, we actually make it, we put in some dry rite in a pouch, seal it, and that's how we store it, and it works really well. So, what about multiplexing it? In principle, we should multiplex it by printing different capture antibodies. So here's an assay. Again, when we do proof of principle, we did one against, printed capture antibodies for IL-6 and TNF-alpha. So what you see here is a dose response curve for TNF-alpha, because I printed capture antibodies against IL-6 and TNF-alpha, but when I printed the detection antibodies, in this case, I only printed the TNF-alpha detection antibody and deliberately skipped the detection antibody for IL-6. So what you can see here is you get a dose response curve for TNF-alpha, but you get a flat line for IL-6, which is what you would want. Otherwise, if you start seeing a dose response curve for this, it means you have cross-reactivity, which is bad. If you flip it, now you get, in this case, sorry, this data is, okay, just look at the top slides, it's the same thing. Then you reverse it. Now you get a dose response curve for IL-6 and not for TNF-alpha by now printing the other detection antibody and skipping the other one. And then when you print both, you get both dose response curves. So this proves that in principle it's possible to print two types of capture antibodies, take two detection antibodies of the same fluorophore, mix them, and get an assay that is not cross-reactive. Okay. But so far everything I've told you has been a bit of a lie, because I didn't tell you how I was imaging the spots. How we were imaging the spots was to go down to the second floor and use the many obsolete microarray scanners at Human Genome, what used to be IGSP, the Institute for Genome Science and Policy. So we now live in this era of next-gen sequencing, PacBio, Illumina. There was an era which the young people have forgotten, where people did Affymetrix, did adjuvant arrays and Affymetrix arrays. You did DNA microarrays, and those DNA microarrays were imaged on this largely on this instrument called the GenePix, which is an 80-pound instrument, $150,000, with a fantastic PMT, great electronics, very sensitive. So you could say, hey, great, you're getting great data because you're cheating. Who's going to carry this to India or Africa or Myanmar? Nobody. It's too expensive, it's too big, it's a beast. So, and this is the kind of data we were getting. Well, the next challenge was building a low-cost detector. I have this amazing graduate student who's only in his second year. He was a Duke undergrad. He went off to industry for 3 months, hated it, came back to my lab as a technician, is now a PhD student. In 2 years, he's built a detector that takes companies many millions to build, which tells you a few good people are all you need in life. You don't need a massive team. In fact, I'll show you the team. It's tiny, my team. So Jason, this is DIY kid. He's a kid. This is what I love about American graduate students. They're really brave, and they do all this stuff in their kids, which I grew up in India. I didn't learn anything useful besides solving differential equations, which turns out to be not that useful in life. So he knows how to 3D print. He taught himself optics. I sent him off to a new faculty member's lab, Roark Horstmeyer, who's the king of optics, and he just sort of absorbed stuff, and he hung out with Roark, and he started building these generations of detectors. Every 3 months was a new generation. So what you do is, the way he built it is, he had a cheap LED laser, costs nothing, a few dollars, that hits at an oblique angle. Then he has a cheap filter, and then he has a camera, and we just buy the Sony camera that everybody uses, the CMOS camera. All these parts you can get off eBay, actually, and you can get them from China. And then he 3D printed the casing for it. So this is what the parts look like. This is the chassis that it's assembled on. So all the chassis is 3D printed. He just snaps it in place and then puts a nice box around it. More details that you need. So the first generation had a big computer. So the main part is you have the LED, some filters, the camera, and the camera is on this device. It's connected to something called a Raspberry Pi computer, which is a very low-cost computer, which we use in BME actually for design. It's open source. You can program it. It's very cheap. It's actually used a lot in engineering education right now. And it has a touch screen. So first he had a big screen and it was kind of big, and then he kind of made some changes. So now our second generation, which he calls the Einstein, is a more user-friendly. It's smaller. It's cheaper. Has longer battery usage, so you can go for 16 hours. And he also wrote software that he programs directly into this Panda Latte, this Raspberry Pi computer, where it will go find the spots. So he's written a little machine vision software, finds the spots, and then calculates the intensity, subtracts the background, and once you put a chip in, within 2 seconds, it'll give you the data. And it'll show up on the screen. It'll show the spots, it'll show the concentration, and this is Wi-Fi compatible, you can send it to a server. So everything can be sent to a server and stored. And it's going to be RedCap compatible because we have to go to China or Guatemala to do some access. So this is what it looks like. It's about this big. It weighs a few pounds. It costs us about $750. And the biggest expense is actually that filter's massively expensive, and then it's the computer and the CMOS sensor. This is buying like 1 or 2 parts at a time. If you were to buy 10,000 of these, I think we can drop the price down to a few hundred or $100 because they're charging retail prices one at a time. And if you go to Sony and say I want 10,000 of these, I think they would give us a better price. But even $750 is not bad. Fine, you can build it, but how does it work? Right? I mean, anybody can build anything. Does it even work, and how well? It works remarkably well. I mean, Jason is amazing. Here's a GeneSpeak scanner. Here's our D4 scope. Dose response curve, identical. Sometimes we're a little bit worse, but we're very, very close, and it's good enough for most things. And concordance, meaning how well does the data agree? R square of 0.996. I mean, you can actually see the concordance right here. Don't need this, but. So I'm going to switch now and show you data on Ebola using this detector. And this is a project, a PhD project of a really amazing student, Cassio Fontes, who actually had a company in Brazil doing diagnostics. They had 50, 60 products in the market. Basically, they copied everything from the US and would just get certified. They were really good at regulatory approvals, is what they were good at. He got bored of what he said. He got bored of like opening up other people's diagnostics and re-engineering them. He actually wanted to learn how to build new stuff. So he found me and he came. And in his PhD, he's built this amazing diagnostic for Ebola, where his goal was to build the most sensitive point of care. Cassio is a man of great ambitions. So he focused on Ebola again, through an accident. This is a collaborative project, I should point out, with D-Gun. So it turns out Ebola is really interesting and has a very interesting life cycle, which I won't go into, but from the perspective of diagnostics, what is interesting is that Ebola infects cells and it hides in there for a while. And while it's hiding, it's doing a lot of sort of trying to fool the body. And one of the things it does to fool the body is to make a protein that it secretes out that is a truncated version of one of the major coat proteins of the viral capsid. So there's a protein called glycoprotein, which is one of the proteins in its capsid. Through some clever RNA editing, transcription splicing, it makes 2 versions. One is a soluble version that's shorter, that actually gets secreted from the cell, dimerizes. Within the first 4 or 5 days of infection, it's at 50 to 100 nanograms per mil concentrations. Huge amounts of this protein. It's an antibody decoy, so that if the body has any antibodies that could bind to the capsid, it's going to bind to this soluble protein and get cleared. Right? So it's keeping the antibody-mediated defense of the body at bay. It also has ways of getting rid of cellular-mediated immunity or attack. And only 20% is actually the transcript that makes the viral capsid. So this is obviously a great marker to go for because the virus is amplifying its presence by sending out this message. It's actually broadcasting it's there, except it's actually using it to fool the body. It was pointed out many years ago by somebody at CDC, whose name I can't remember, that this would be a great marker to go for. Somehow, for some reason, the field ignored him, and he retired a somewhat embittered man because when we contacted him for his antibody stocks against SGP, he said, oh, threw it all out. Nobody cared it. So D-Gun did some phage display, and basically, there are many ways of making antibodies, and we worked with him in screening it, and we found a pair that binds to SGP, because these antibodies are not commercially available. So we now have a really good pair against soluble glycoprotein made in-house at Duke through D-Gun's help. And I will show you now a D4 assay where we're going to use these 2 antibodies. So the first thing we're taking advantage of is the fact that the virus is doing amplification. And I use the word amplification because what we're going up against is PCR, right? So the most sensitive point of care test for Ebola is PCR, which is kind of crazy. They have to set up these tents. Everybody wears gloves and masks. PCR is very sensitive, but also sensitive to contamination. And you have to train people how to do this. It just seems crazy. The other test that's available on the other end is a lateral flow assay, which has horrible sensitivity. I won't show you the data, but we've actually built our own LFAs. Our assay is like 100 to 1,000 times more sensitive. But it's approved, but it diagnoses it very late, by which point people are so sick they're going to die. And now that we have antibodies available against Ebola, it turns out the sooner you diagnose, that antibody that Tony Fauci just published works. So it used to be, why do you care about early diagnosis? I mean, if you think about it practically, it used to be early diagnosis, you could sequester people, quarantine people, prevent the spread of disease, because you get more and more infectious terms of transmission as the disease progresses. In fact, if you catch people early, Ebola is not transmissible. So I didn't know this. But now there's an even more important reason, which is not to just prevent the spread of disease. If you can catch people in the first 4 days of infection, the cure rates skyrocket. I mean, like, it's amazing. You can get 80% cures if you do it by day 3 or day 4. It drops to 20% by day 7. So catching people early is really great because you can finally cure Ebola. If you have the antibody drug, but early detection is key. So here's our assay for SGP. I'm going to skip through the details because you've already seen the assay. Here's what the dose response curve on the gene picks on this big beast. Here's on ours. It varies. I mean, in this case, we're not better. This is the same number. But we're not better. We're about the same. And here's some data which is sort of plotted weirdly, so I'll apologize. So we're looking at fluorescence intensity, days post-infection. This is data from macaques. These are monkeys that are being infected at Galveston at a BSL-4 lab. I can say that no monkeys died because of us. They were going to die anyway because they were doing experiments at Galveston is where a lot of the BSL-4, the Ebola work gets done. So we piggybacked on some existing experiments and they were very kind to take blood samples for us. So these are the control animals. These are animals that were exposed to Ebola, either they scored it in the eye or they do it intramuscularly, and then they give them different drugs, prophylactics, vaccines. We just asked them to take blood from the control animals. Okay, so we piggyback on experiments where from the control animals that are infected, we get blood samples and they do the analysis. So this detector actually has been sitting in Galveston for months now because we went there, or Cassio, Jason went there, trained the technician Crystal, and then the lab goes hot, she takes everything in, and then we just get emails and we get data streams from Galveston. So the data is generated by her, which is great. We now have at least highly trained technicians. And if you like Galveston, those technicians are amazing. You have to be to work in a BS4 facility, but she uses this test all the time. So we have our first beta tester. And so the limit detection for Ebola is sort of here. This is of our assay, okay, from the dose response curves. This is day 0. Day 1, we can't see anything. At day 2, we could pick out 2 out of 4 monkeys. We could detect it, okay? By day 6, everything starts to work. LFA, PCR, and RT4 assay. By day 3, PCR only works on 1 animal. So we get 2 animals here out of 4. By day 4, we get 100% sensitivity. We can catch all the Ebola infected monkeys. But PCR only catches 1. And then the PCR starts to work somewhere by day 5. So our threshold is day 3 to day 4 in terms of sensitivity specificity. I mean, we haven't done enough animals to say sensitivity specificity, but 2 out of 4, 100%. So 50%, 100%, they're at 33, PCR at 33, and then 100% the next day. So we are at least a day better, and we've seen this both for conjunctival and for intramuscular routes of infection. And why are we better? Because soluble GP is doing the amplification for us, and then we are marrying it to an extremely sensitive assay. And you can see, here's the actual assay. You can see how much the level of SGP skyrockets in a few days. This had been published before. This is our own data from these monkeys. So you can see, day 0, day 1, day 2, you start to see SGP being pumped out. By day 3, it's swamping the system. How much time do I have? I don't want to go on too long. I have a few more slides.
10 more minutes.
Okay, I'll take 3 minutes and be done. So the Ebola work is actually unfunded. We sent in an RO1 and we got hammered. We went to an engineering study saying, like, why do you need this? Why is this better? We have LFA, we have PCR, who cares? So what if you catch them early? Anyway, it was kind of depressing. We thought we had great data, we're beating PCR, come on, fund us. But they didn't fund us. But we are funded by the NCI for a hepatocellular project, HCC project, for a cancer project, which one might actually argue deserve less funding because there are very, very few good markers for cancer. And I've always been, I see a clear need for infectious diseases. That's like with cancer, the argument gets a little weaker, but NCI has something called Global Cancer. They do RFAs, they fund people, they were kind enough to fund us, so we have a U grant from them. But the idea was, they said, we don't care, pick any cancer and give us a point of care assay. So we looked around, we didn't know anything about it. We found hepatocellular carcinoma, because it's widely distributed across the world. We had to work with one of 30 or 40 countries, there are LMIC countries. And China has a lot of it, so we decided to work in China, which is another story I'll tell you about later, how difficult it is turning out to work in China. In fact, we try to pull out of China right now. I can't deal with it anymore. Trying to move to Guatemala, because Guatemala also has a lot of HCC. So, it's sort of interestingly distributed. There's been a bunch of papers showing that you should be able to sort of get some sort of diagnosis of HCC, hepatocellular carcinoma, using serum markers. So we went through and looked at all the different papers, and we found six biomarkers that different papers have implicated in either diagnosing between HCC and healthy or at risk in HCC and so on. As you'll see, these markers are all terrible. In fact, when you combine them, they sort of start to work. What they are isn't important. So we built a chip for all 6 markers, and this is quite a tedious process because we buy every antibody in the market against that marker, and then we pair them using a microarray, which speeds it up, and then we find the perfect pair. So we do a whole pairing exercise where we try and get every antibody that exists against DKK3 or alpha-fetoprotein. That gives us the best pair, and here's the assay we built. So here's a low concentration sample. We can barely see the spots. You can see it in my computer, but not here. Here's a high concentration sample. So we built this 6 analyte array. Here's a dose response curve. The question is, can you actually build a 6 analyte array without cross-reactivity? Yes, you can. This is part of our whole screening exercise to find antibody pairs that are highly sensitive and not cross-reactive with any of the other antibody analyte combinations. This takes, if you do it at ELISA, this will take you years. We use a microarray, this takes us like a month. And so here's the dose response curve for all 6. The data is not important. What is important is looking at the concordance between the filled and open spots. And you can see multiplex versus single analyte, multiplex versus single analyte, they pretty much lie on top of each other. So we have a 6-plex array for HCC markers, which are serum markers, protein markers that are non-cross-reactive. And when you start looking at patient samples, so Duke only has a few HCC samples, and then at-risk means people who have cirrhosis, fibrosis, had been diagnosed with hepatitis B, hepatitis C. You lump them all together, and you call them at risk. We got some samples from Duke, then there are these companies that sell these, I don't know, not very well curated samples. They claim they have them, so we just bought a whole bunch, and we did this. We looked at patient samples, archival, and you can see how terrible these markers are, right? So maybe DCP has some value, maybe AFP does, maybe this one does. This was supposed to be actually a good one. Anyway, we take all these 6, this looks like noise to me, and you send it to a statistician, and you get an ROC, an area under the curve, where 100% sensitivity specificity would be, just 1/2 of a rectangle, and flipping a coin is a diagonal. So you want to be not a diagonal, you want to be as close to a square wave function, is the way to explain it. And that gives you the area under the curve. You want a high area under the curve. So we can ACO 0.94 and 0.86 for these samples. We since then have looked at 100 patient sample from NCI, and we are still bitterly debating how the data should be analyzed. They don't want to tell us anything about it, but they also don't want to analyze it de novo. They want to use our model, and we're trying to explain to them the whole point of this millions of dollars of funding is for us to build the test, get hundreds of patients and samples, and then build a statistical model, right? Because really what you're doing here is you're taking all this data and you're doing a multivariate regression model to get this area of the curve. And they said, no, give us a model. I said, my model is useless. I'm only halfway through the project, and this is very obdurate statistician at Fred Huck, but he's like the most important guy at NCI, and you can't talk to him. He just says the same thing back, and he's like, no, you must have a model. It's your data, you build it, and tell us how data looks anyway. So this is going to go on for years at this point, and NCI cannot seem to mediate this argument. So I'm going to stop. So anyway, the bottom line is, it works, it sort of works. We'll see how it works. This project is not supposed to go to China, but it's been impossible to work in China lately, so we have permission from NCI to pull out potentially and go to Guatemala, because it also turns out NCI intramural works in Guatemala and Mexico. So we can piggyback on these amazing, great relationships that intramural NCI epidemiologists, liver cancer specialists have in Guatemala, and at least the quality of interactions between China and Guatemala have been remarkably different. I mean, what I've learned is you better pick your global site partners really carefully. You're really naive, right? Now we're learning, oh my God, it's like night and day. I mean, the Guatemalans actually know how to work with the NCI system. They already have red cap set up. They know how to fill out all the right forms. It's like pleasure. So it's not that China is necessarily bad. It's that the place we went to, we just had a lot of trouble. So I'll stop here. Hongwei Ma was a student who developed the brushes and then made a terrible assay, but that's not his fault. He'd done a lot of great work by that time. Angus Hucknall is really the guy who made everything possible. So Angus developed the first generation array, the femto array, and the D4 POCT We got on the femtoray because we actually started a company, and I like coming up with trade nameable names. We actually sold the company to a company named Immucor in 2014. Now we're trying to buy it back from them, which is another story. Then Daniel Joe was an MD PhD student who did a lot of clinical applications, took us, that's a great thing when MD PhD students, they really take you into the hospital. Cassio did the Ebola work and much else. He's a senior graduate student now. So the team now is really Angus and Cassio working with this amazing crew here, where I call Jake the chip guy. So he built the chip for HCC. That's the most sophisticated chip we've built. I didn't talk about the microfluidics work we're doing. That's David. And then Jason is the guy with the detector. Roy Horstmeyer for detector design. A lot of help. T-Gun for the antibodies. And Thomas Geisbert and his crew at UT Galveston for the monkey studies in Ebola. Lots of funding sources. What has been interesting is that my first half of my career, nobody wanted to fund this diagnostics work. They're like, this is boring, who cares? And now everybody wants to fund it because global health has become fashionable. I mean, it really has. I mean in a good way. And NIH puts a lot of money into global health. I get no money from the Gates Foundation. I get all my money from from NIH and oddly enough, Department of Defense, because for them, global health is low cost devices that can be given to special forces. Right? This is what. So actually, one of our first grants came from the special forces, believe it or not. So there's sort of a good. There's a good and dark side to all of this. And right now we're negotiating a massive grant from something called the Medical MCBRN, which is part of DITRA, which is part of something else. All these acronyms, DoD acronyms, and they really want us to build a point-of-care assays for cells, bacteria, viruses, proteins, for things like the plague, Ebola, anthrax, et cetera, things that I hope nobody will ever be infected with in the US, but DoD is interested in. We take their money because we're agnostic. It's US taxpayer money and it develops technology, and our hope is to save lives. And if you save the lives of US warfighters, that's fine too. Thank you for your attention, and I'd be glad to take questions.